आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सोल्यूशन पाओ। डाउनलोड नाउ।
In this question, they are asking what is semi-conservative DNA replication and how it was experimentally proved. Now we all know that DNA replication is a process through which DNA generates its own copies or duplicates itself. Now if we look at the structure of DNA, according to the double helix model that has been given by Watson and Crick, the DNA is made up of polynucleotide chains. Polynucleotide is nothing but a polymer of repeating units known as nucleotides. And we see that our DNA is a double-stranded structure, which means that two polynucleotide chains, which are anti-parallel to each other or which are complementary to each other, come together and are joined together by strong hydrogen bonds, thus forming the double helix DNA. Now, as soon as they gave us the double helix model, Watson and Crick also suggested a copying mechanism for the replication of DNA, and they guessed that out of both of these strands of the DNA, we are going to see that they act as templates for the replication of DNA for the synthesis of complementary strands. But whenever there is a newly formed DNA, we are going to find that this DNA replication is semi-conservative, which means that suppose a double-stranded DNA is being replicated, then in the newly formed DNA, one of the strands, one of the polynucleotide chains is going to be newly synthesized, whereas the other one would be the parental or the template strand. So this copying mechanism, which has been suggested by Watson and Crick, is known as semi-conservative DNA replication, which means any DNA molecule, any double-strand DNA is going to contain one of the newly synthesized and one of the old parental strands. Now, this was experimentally proven by Matthew Meselson and Franklin Stahl in 1958. During this experiment, they worked with the E. coli bacteria, and what they did was they grew this E. coli on a medium containing ammonium chloride, but in this ammonium chloride, the nitrogen was not the normal nitrogen, it was the heavy isotope N15. So what happened was, whenever this E. coli would divide, this heavy nitrogen is getting incorporated into the DNA of the E. coli. So after all of the E. coli DNA had become heavy, after this heavy isotope had been incorporated into it, now this E. coli bacteria was removed from this medium and placed in the medium where we had the normal hydrogen isotope. So suppose it is believed this E. coli is going to divide every 20 minutes. So what we saw after 20 minutes was that now the DNA which has been formed after replication of E. coli, it is a hybrid type of DNA. Hybrid means that in the double-strand DNA molecule, one of the strands is containing the heavy isotope, whereas the other strand is normal. So this proves that when DNA is being replicated, when DNA is being copied, one of the strands is the old one, and one of them in the double-strand molecule is the newly synthesized one. Similarly, after 40 minutes, we observed that once again DNA has been replicated, and half of the E. coli bacteria contain hybrid DNA and the rest of them had normal DNA, because now there was no N15, there was no heavy isotope in the medium on which they were growing. So this experiment conducted by Meselson and Stahl was the experimental proof for semi-conservative DNA replication.
क्लास 6 से 12 से लेके नीट, IIT, जेईई मेंस और एडवांस के लेवल तक 10 मिलियन से ज्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।